Hello students, today we will talk about Immunochemical Techniques 3. Introduction. All vertebrates have advanced immune system of their body. The more complex the organism, the more advanced the immune system. The immune system of mammals has evolved over 1 million challenges that arise in the body. Immunity in our body is monitored or controlled by specific cells from stem cells in bone marrow. The most important cell types are B and T lymphocytes, which have the ability to act against bacterial and other viruses. B cells release antibodies against stimulation of a foreign substance into the body. An antigen is a foreign substance capable of an immune response leading to the production of antibodies. They are the targets to which antibodies bind. So, antibodies are antigen specific. That is, it binds to the antigen that initiated its production. This chapter focuses on the detection and analysis of the specific antibody or antigen present in the sample using complement fixation test and Western blotting technique. First, complement fixation test, in short CFT. Coming to the principle. Complement is the kind of protein present in normal body serum that can be activated directly by pathogens or indirectly by pathogen-bound antibody, leading to a cascade of reactions that occurs on the surface of pathogens and generates active weight components with various effective function. This whole cascade pathway is termed as complement system, and it is made up of 9 protein components, C1 to C9. Complement binds to antigen-antibody complex. When antigen forms a complex with antibody on surface of a cell, complement causes lysis of cell. CFT is a technique that has been used over many years to detect and quantify antibody that serology does not agglutinate or precipitate when reacted with its antigen, but can be demonstrated by its use or fixation of complement. Antigen-antibody reactions lead to immune complex formation that produces complement fixation via the classical pathway. That is when complement takes part in antigen-antibody reaction. It is bound or fixed to the antigen-antibody complex. When this complex is on the bacteria, red cells or other cells, the complement brings about the lysis of cells involved. This can be exploited to determine the amount of antigen-antibody present in the patient's serum. CFT can detect antibody at a level of less than 1 microgram per milliliter. This picture shows the positive test and negative test of complement fixation test. Complement fixation test require 2 steps. Step 1. A non-antigen and inactivated patient serum are incubated with a standardized limited amount of complement. If the serum contains specific complement activating antibody, the complement will be activated or fixed by the antigen-antibody complex. However, if there is no antibody in the patient's serum, there will be no formation of antigen-antibody complex, and therefore complement will not be fixed but will remain free. The second step detects whether complement has been utilized in the first step or not. This is done by adding the indicator system. If the complement is fixed in the first step owing to the presence of antibody, there will be no complement left to fix to the indicator system. However, if there is antibody in the patient's serum, there will be no antigen-antibody complex, and therefore complement will be present free or unfixed in the mixture. This unfixed complement will now react with the antibody-coated SIP red blood cells to bring about their lysis. Thus, if no lysis of SIP red blood cells takes place, then it is said to be positive CAFT, which indicates the presence of antibody in the test serum, while lysis of SIP red blood cells will give negative CAFT, which indicates the absence of antibody in the serum. Let's come to the advantage of CFT. Number 1. Ability to screen against a large number of viral and bacterial infections at the same time. Number 2. It is not very expensive. Then next disadvantage of CFT. Number 1. Not sensitive, cannot be used for immunity screening, time consuming and labor intensive, often non-specific, example cross-reactivity between herpes simplex virus and varicella zoster virus. Modification of complement fixation test. A. Indirect complement fixation test. This modification is used when serums which don't fix guinea pig complement is to be tested. Here, the test is set up in duplicate. After step 1, standard antiserum to antigen which is known to fix complement is added to one set. If antibodies were not present in the test serum, then the antigen would react with the standard antiserum fixing the complement. On the other hand, if antibodies are present in the test serum, the antigen would be utilized in the first step. So, no reaction would occur between the standard antiserum and antigen, and therefore no fixation of complement would cause lysis of SIP red blood cells. Thus, in this case, hemolysis indicates a positive result. Number B. Coagulating complement absorption test. Here, horse complement, which is non-hemolytic, is used. The indicator system used in sensitized SIP red blood cells mixed with bovine serum. Bovine serum contains a beta globulin called coagglutinin would also combine with the complement causing agglutination of the SIP red blood cells, indicating a negative result. Number C. Immune adherence. When some bacteria such as Vibrio cholera or Treponema pallidum combine with their specific antibody in the presence of complement and some particles such as erythrocytes or platelets, they adhere to the erythrocytes or platelets. This is called immune adherence. Number D. Immobilization test. Here, antigen is incubated with patient serum in presence of complement. If specific antibody is present, it would immobilize the antigen. Example, Treponema pallidum immobilization test, considered gold standard for the serodiagnosis of syphilis. Number E. Cytolytic test. The incubation of a live bacterium with its specific antibody in the presence of complement leads to the lysis of the bacterial cells. This is the basis of virucidal antibody test used to measure anti-cholera antibodies. Next, coming to the Western blotting. Western blotting is also known as immunoblotting. It is a well-established and widely used technique for the detection and analysis of proteins. The method is based on the building an antibody-protein complex via specific binding of antibodies to proteins immobilized on a membrane and detecting the bound antibody with one of the several detection methods. The Western blotting method was first described in 1979 and has since become one of the most commonly used methods in life sciences research. Coming to the working principle, in Western blotting, a protein mixture is electrophoretically separated on an SDS polyacrylamide gel, a slab gel infused with sodium dodecyl sulfate. Then protein bands are transferred to a nylon membrane by electrophoresis, and the individual protein bands are identified by flooding the nitrocellulose membrane with radiolabeled or enzyme-linked polyclonal or monoclonal antibodies specific for the protein of interest. Steps involved in the Western blotting. Although the details of Western blotting protocols may vary from application to application, with adaptation to suit specific protein characteristics and the level of information required. They all followed some common basic steps. Number 1. Sample preparation. All sorts of protein, from single cells to whole tissues as well as extracellular matrices, biological fluids, and the proteins secreted in vitro are open to analyze by Western blotting. One should follow a popular and particular extraction method, which is suitable with the source. At the same time, obtaining a sufficient yield of material at an acceptable level of purity is very important. And this is the table showing overview of extraction options for different cells and tissues. Number 2. Gel electrophoresis. Electrophoresis is a commonly used method for separating proteins on the basis of size, shape, and/or charge. Polyacrylamide and agarose gels are the most commonly used matrices in research laboratories for separation proteins and nucleic acids respectively. And this is SDS polyacrylamide gel electrophoresis apparatus. And next is the protein bands seen after SDS polyacrylamide gel electrophoresis. Number 3. Transfer of protein bands. On completion of the separation of proteins, the next step is to transfer the proteins from the gel to the solid support membrane, usually made up of chemically inert substance such as nitrocellulose or polyvinylidene difluoride. Blotting makes it possible to detect the proteins on the membrane using specific antibodies. The proteins transferred from the gels are immobilized at the respective relative migration positions at the time point when the electric current of the gel run was stopped. It is important that transfer is as uniform as possible across the entire area of the gel membrane sandwich in order to ensure that large and small proteins are transferred with similar efficiency. And this picture shows transfer of protein bands from SDS page to immobile nitrocellulose membrane. Then number 4, blocking. Blocking of non-specific binding is achieved by placing the membrane in the dilute solution of protein, typically bovine serum albumin (BSA) or non-fat dry milk in trace buffer saline with a minute percentage of detergent such as Twin 20 or Triton X 100. The protein in the dilute solution attaches to the membrane in all places where the target proteins have not attached. Thus, when the antibody is added, there is no room on the membrane for it to attach other than on the binding sites of the specific target protein. This reduces background in the final product of the Western blot, leading to clearer results and eliminates false positives. Next, number 5, primary antibody. After blocking, a dilute solution of primary antibody, which is generated when a host species or immune cell culture is exposed to the protein of interest, is incubated with the membrane under gentle agitation. Typically, the solution is composed of buffer saline solution with a small percentage of detergent. The antibody solution and the membrane can be sealed and incubated together for anywhere from 30 minutes to overnight. Next, number 6, secondary antibody. The unbound primary antibodies are removed by rinsing and the membrane is exposed to another antibody directed at a species-specific portion of the primary antibody. Antibodies come from animal sources or animal source hybridoma cultures. The secondary antibody is usually linked to radioactive level probe, fluorescent level, or a reporter enzyme such as alkaline phosphatase or horseradish peroxidase. Enzyme-linked secondary is used to cleave a chemiluminescent agent, and the reaction product produces luminations in proportion to the amount of protein. Next, number 7, detection. A variety of detection systems based on chemiluminescence, fluorescence, chromogenic, or radioisotopic detections are available. Let's come to the chemiluminescence detection. Chemiluminescence detection methods depend on incubation of the Western blot with a substrate that will luminance when exposed to the reporter on the secondary antibody. The light is then detected by CCD cameras which capture a digital image of the Western blot or photographic film. The use of film for Western blot detection is slowly disappearing because of non-linearity of the image, non-accurate quantification. The image is analyzed by densitometry, which evaluates the relative amount of protein staining and quantifies the result in terms of optical density. Newer software allows further data analysis such as molecular weight analysis if appropriate standards are used. And this figure shows the principle of chemiluminescence detection in Western blotting. Next, radioactive detection. Radioactive levels allow the placement of medical X-ray film directly against the Western blot, which develop as it is exposed to the level and create dark regions which correspond to the protein bands of interest. The importance of radioactive detection method is declining due to the hazardous radiation because it is very expensive, health and safety risks are high, and enhanced chemiluminescence provides a useful alternative. Next, fluorescent detection. The fluorescently level probe is excited by light and the emission of the excitation is then detected by a photosensor, such as a CCD camera equipped with appropriate emission filters which captures a digital image of the Western blot and allows further data analysis such as molecular weight analysis and a quantitative Western blot analysis. Fluorescence is considered to be one of the best methods for quantification, but is less sensitive than chemiluminescence. And this is the figure of proteins which are detected by fluorescence detection method. Coming to the conclusions. In this chapter, we have learned the principles and the working of complement fixation test and Western blotting. These two methods are widely used in the fields of molecular biology, biochemistry, immunogenetics, and other biology disciplines. They played a central role to confirm the presence of target proteins. Western blotting is the most widely used procedure in confirmatory testing for HIV, where it is used to determine whether the patient has antibodies that react with one or more viral proteins. Thank you.